Tonight's lecture is over Chapter 25, Metabolism and Nutrition. Chapter 25 begins on page 1000 in your textbook. Metabolic reactions contribute to homeostasis by harvesting chemical energy from consumed nutrients for use in the body's growth, repair, and normal functioning. Metabolism refers to all of the chemical reactions that occur in the body. There are two types of metabolism, catabolism and anabolism. Those chemical reactions that break down complex molecules into simpler ones are collectively known as catabolism or catabolic. Overall, catabolic reactions are termed exergonic. In other words, they produce more energy than they consume by releasing the chemical energy stored in organic molecules. Furthermore, important sets of catabolic reactions occur in glycolysis, the Krebs cycle, and the electron transport chain. On the other hand, chemical reactions that combine simple molecules and monomers to form the body's complex structural and functional components are collectively known as anabolism or anabolic. Examples of anabolic reactions are the formation of peptide bonds between amino acids during protein synthesis, the building of fatty acids into phospholipids that form the plasma membrane bilayer, and the linkage of glucose monomers to form glycogen. Anabolic reactions are termed endergonic because they consume more energy than they produce. Metabolism results from the balance of anabolic and catabolic reactions. The molecule that most often performs this task is ATP. ATP consists of an adenine molecule, a ribose molecule, and 3 phosphate groups bonded to one another. This is on page 1001. This is figure 25.1. This shows the coupling of energy-releasing and energy-requiring reactions that can be achieved through ATP. When complex molecules and polymers are split apart in catabolism, some of the energy is transferred from ATP and the rest is given off as heat. When simple molecules and monomers are combined to form complex molecules, as in the case of anabolism, ATP provides the energy for synthesis, and again, some energy is given off as heat. So the fact that heat is released on both of these reactions just goes hand in hand with the laws of thermodynamics. And that is, energy cannot be created or destroyed. And secondly, for any energetic reaction, there's always a net loss of energy. And in this case, the energy is lost as heat. And that is part of the reason why you have body heat. Moving on to the next slide. This is on page 1002, this little chemical equation that you see in front of you. Oxidation-reduction reactions are one category of reactions that is important for the transfer of energy. Oxidation involves the removal of electrons from an atom or a molecule. And in this example, we see the conversion of lactic acid to pyruvic acid. And then reduction involves the addition of electrons to a molecule. And here we see the conversion of pyruvic acid to lactic acid. One way you can keep these two reactions straight is through an acronym. That acronym is OIL-RIG. Oxidation is a loss, reduction is a gain. When a substance is oxidized, the liberated hydrogen atoms do not remain free in the cell, but they are transferred immediately by coenzymes to another compound. Two coenzymes that are commonly used by animal cells to carry hydrogen atoms are nicotinamide adenine dinucleotide. Sometimes we refer to that as NAD. This is a derivative of the B vitamin niacin. And then flavin adenine dinucleotide, or FAD. And this is a derivative of vitamin B2, or riboflavin. The oxidation and reduction states of NAD plus and FAD can be represented in the following equations. So if we look at the first reaction, when NAD plus is reduced to NADH plus a hydrogen ion, the NAD plus gains the hydride ion. This neutralizes its charge and the hydrogen ion is released into the surrounding solution. When NADH is oxidized to NAD+, the loss of the hydride ion results in one less hydrogen atom and an additional positive charge. In the other equation, FAD is reduced to FADH2 when it gains a hydrogen ion and a hydride ion. And FADH2 is oxidized to FAD when it loses the same two ions. Oxidation-reduction reactions, or redox reactions, are what we call a coupled reaction. They're coupled because anytime one substance is oxidized, another substance is reduced, or vice versa. And we can see that in this equation on the slide we're looking at. An important point to remember about oxidation-reduction reactions is that oxidation is usually exergonic, or energy-releasing. Cells use multi-step biochemical reactions to release energy from energy-rich, highly reduced compounds to lower-energy, highly oxidized compounds. For example, when a cell oxidizes a molecule of glucose, the energy in the glucose molecule is removed in a stepwise manner. Ultimately, some of the energy is captured by transferring it to ATP, which then serves as an energy source for energy-requiring reactions within the cell. Compounds with many hydrogen atoms, such as glucose, contain more chemical potential energy than oxidized compounds. For this reason, glucose is a very valuable nutrient. Moving on to the next slide. Some of the energy released during oxidation reactions is captured within a cell when ATP is formed. A phosphate group is added to ADP with an input of energy to form ATP. And if you notice the squiggly lines on the slide, those show the bonds that can be used to transfer energy. So the high-energy phosphate bond that attaches the third phosphate group contains the energy stored in this reaction. The addition of a phosphate group to a molecule is called phosphorylation, and it increases its potential energy. Organisms use three mechanisms of phosphorylation. Those three mechanisms are substrate-level phosphorylation, oxidative phosphorylation, and photophosphorylation. In substrate-level phosphorylation, ATP is generated by transferring a high-energy phosphate group from an intermediate phosphorylated metabolic compound. In oxidative phosphorylation, electrons are removed from organic compounds and they are passed through a series of electron acceptors called the electron transport chain to molecules of oxygen. And in photophosphorylation, chlorophyll-containing plant cells or bacteria contain light-absorbing pigments. Carbohydrate metabolism. Carbohydrate metabolism is mostly glucose metabolism. The body's use of glucose depends on the needs of the cell. These needs include ATP production, amino acid synthesis, glycogen synthesis, and triglyceride synthesis. Concerning ATP production, In body cells that require immediate energy, glucose is oxidized to produce ATP. Glucose is not needed for immediate ATP production, can enter one of several metabolic pathways. Concerning amino acid synthesis, cells throughout the body can use glucose to form several amino acids, which then can be incorporated into proteins. In glycogen synthesis, hepatocytes and muscle fibers can perform glycogenesis, in which hundreds of glucose monomers are combined to form the polysaccharide glycogen. The total storage capacity of glycogen is about 125 grams in the liver and about 375 grams in skeletal muscles. And finally, concerning triglyceride synthesis, when the glycogen storage areas are filled up, hepatocytes can transform the glucose to glycerol and fatty acids that can be used for lipogenesis. That's the synthesis of triglycerides. Triglycerides are then deposited in adipose tissue, which has virtually unlimited storage capacity. Moving on to the next slide, glucose must pass through the plasma membrane to be used by the cell, and this requires facilitated diffusion. Glucose entry into most of the body cells occurs via GLUT molecules. This is a family of transporters that bring glucose into cells via facilitated diffusion. A high level of insulin increases the insertion of one type of GLUT, called GLUT4, into the plasma membranes of the body cells, thereby increasing the rate of facilitated diffusion of glucose into the cells. In neurons and hepatocytes, however, another type of GLUT is always present on the plasma membrane, so glucose entry is always turned on. On entering a cell, glucose becomes phosphorylated. Because GLUT cannot transport phosphorylated glucose, the reaction traps glucose within the cell. Next slide. The oxidation of glucose to produce ATP is called cellular respiration, and 4 sets of reactions are involved in cellular respiration. That is glycolysis, the formation of acetyl coenzyme A, the Krebs cycle reaction, and the electron transport chain reactions. So this is figure 25.2 on page 1004, and in this figure we see an overview of cellular respiration. So the first step of cellular respiration is glycolysis, in which a set of reactions in one glucose molecule is oxidized into 2 molecules of pyruvic acid. The reactions also produce 2 molecules of ATP and 2 energy-containing NADH molecules. Step 2 is the formation of acetyl coenzyme A, and this is a transition step that prepares pyruvic acid for entry into the Krebs cycle. This step also produces energy-containing NADH plus carbon dioxide. The third reaction is the Krebs cycle. These reactions in the Krebs cycle oxidize acetyl coenzyme A to produce carbon dioxide, ATP, NADH, and FADH2. And finally, the electron transport chain reactions. In the electron transport chain reactions, NADH is oxidized and FADH2 is oxidized. This transfers their electrons through a series of electron carriers that will ultimately find their way to an oxygen molecule and create water. So this is why I always say that the respiratory system and the urinary system are intimately entwined. And this is why we need to inhale oxygen. So moving on to the next slide and going a little bit more in detail about glycolysis. Glycolysis is the process whereby a 6-carbon glucose molecule is split into 2 3-carbon molecules of pyruvic acid. Glycolysis actually involves 10 reactions. So this next slide is figure 25.3 on page 1005. So in the first 5 steps of glycolysis, energy in the form of ATP is invested, and the 6-carbon glucose is split into 3 carbon molecules of glyceraldehyde 3-phosphate. And in steps 6 through 10, the 2 glyceraldehyde 3-phosphate molecules are converted to 2 pyruvic acid molecules, and then ATP is generated. This next figure is on page 1006, and this is figure 25.4. And this goes into detail of the 10 glycolytic reactions. So at step 1, glucose is phosphorylated using a phosphate group from an ATP molecule to form glucose 6-phosphate. Secondly, glucose 6-phosphate is converted to fructose 6-phosphate. At the third step, a second ATP is used to add a second phosphate group to fructose 6-phosphate to form fructose 1,6-biphosphate. At steps 4 and 5, fructose splits into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. At step 6, oxidation occurs as 2 molecules of NAD+, accept 2 pairs of electrons and hydrogen ions from 2 molecules of glyceraldehyde 3-phosphate to form 2 molecules of NADH. Body cells use the 2 NADH produced in this step to generate ATP in the electron transport chain. The phosphate group attaches to the glyceraldehyde 3-phosphate, forming 1,3-bisophosphoglyceric acid. Then at steps 7 through 10, these reactions generate 4 molecules of ATP and produce 2 molecules of pyruvic acid. Moving on to the next slide, what happens to the pyruvic acid depends on the availability of oxygen. If oxygen is scarce, in other words, if the condition is anaerobic, pyruvic acid is reduced by the addition of 2 hydrogen atoms to form lactic acid. On the other hand, if oxygen is plentiful, in other words, we have an aerobic condition, most cells convert pyruvic acid to acetyl coenzyme A. So this cartoon is on page 1007. This is figure 25.5, and it outlines the fate of pyruvic acid that we were talking about on the previous slide. So when oxygen is plentiful, we see that pyruvic acid enters the mitochondria and is converted to acetyl coenzyme A. Then it enters the Krebs cycle. This is called the aerobic pathway. On the other hand, when oxygen is scarce or we have an anaerobic condition, most of the pyruvic acid is converted into lactic acid, and this is called the anaerobic pathway. This cartoon is on page 1008. This is 25.6. So we see that after the formation of acetyl coenzyme A, the next stage of cellular respiration is the Krebs cycle. And reactions of the Krebs cycle occur in the mitochondrial matrix. Moving on to the next slide, the Krebs cycle is also known as the citric acid cycle. And this cycle occurs in the matrix of the mitochondria and consists of 8 reactions. And this cartoon is on page 1009. This is figure 25.7, and it outlines the 8 reactions of the Krebs cycle. So step 1 is where the acetyl coenzyme A enters the Krebs cycle, and then the chemical bond that attaches the acetyl group to coenzyme A breaks, and the 2-carbon acetyl group attaches to a 4-carbon molecule of oxaloacetic acid to form a 6-carbon molecule called citric acid. At step 2, isomerization occurs. In this step, citric acid undergoes isomerization to isocitric acid, which has the same molecular formula as citrate. Notice, however, that the hydroxyl group is attached to a different carbon. Step 3 is called oxidative decarboxylation. In this step, isocitric acid is oxidized and loses a molecule of carbon dioxide, forming alpha-ketogluteric acid. The hydrogen ion in the oxidation is passed on to NAD+, which is reduced to NADH. Step 4 is also a step of oxidative decarboxylation. In this step, the alpha-ketogluteric acid is oxidized. It loses a molecule of carbon dioxide and picks up a coenzyme A to form succinyl-CoA. Step 5 is called substrate-level phosphorylation. In this step, coenzyme A is displaced by a phosphate group, which is then transferred to guanosine diphosphate, or GDP, to form guanosine triphosphate. GTP can donate a phosphate group to ADP to form ATP. Step 6 is dehydrogenation. Succinic acid is oxidized to fumaric acid as two of its hydrogen atoms are transferred to the coenzyme flavin adenine dinucleotide, or FAD, which is then reduced to FADH2. Step 7 is referred to as hydration. The fumaric acid is converted to malic acid by the addition of a molecule of water. And step 8 is another dehydrogenation step. In this final step of the cycle, malic acid is oxidized to reform oxoacetic acid. Two hydrogen atoms are removed and one is transferred to the NAD+, which is reduced to NADH. The regenerated oxoacetic acid can combine with another molecule of acetyl-CoA, and this will begin a new cycle. So the three main results of the Krebs cycle are the production of reduced coenzymes, NADH and FADH2, which contain stored energy, the generation of GTP, a high-energy compound that is used to produce ATP, and the formation of carbon dioxide, which is transported to the lungs and exhaled. Moving on to the next slide, the electron transport chain is a series of electron carriers in the mitochondria. Each carrier in the chain is reduced as it picks up electrons and oxidized as it gives up electrons. Exergonic reactions release energy used to form ATP. This mechanism links chemical reactions with the pumping of hydrogen ions known as chemiosmosis. This is figure 25.8 on page 1010. And in chemiosmosis, ATP is produced when hydrogen ions diffuse back into the mitochondrial matrix. So in step 1 of chemiosmosis, energy from NADH passes along the electron transport chain and is used to pump hydrogen ions from the mitochondrial matrix into the space between the inner and outer mitochondrial membranes. This mechanism is called a proton pump because hydrogen ions consists of a single proton. In step 2, a high concentration of hydrogen ions accumulates between the inner and outer mitochondrial membranes. And then in step 3, ATP synthesis occurs as hydrogen ions flow back into the mitochondrial matrix through a special type of hydrogen ion channel in the inner membrane. Moving on to the next slide, electron carriers include flavin mononucleotide. This is a flavin protein derived from riboflavin, or vitamin B2. Cytochromes are another example of an electron character. These are proteins with an iron-containing group capable of existing alternately in a reduced form of an iron ion and an oxidated form of an iron ion. The cytochromes involved in the electron transport chain include cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. Another electron carrier is the iron-sulfur carriers. These contain either 2 or 4 iron atoms bound to sulfur atoms that form an electron transfer center with the protein. Also, copper atoms. Copper atoms are bound to 2 proteins in the chain and also participate in electron transfer. And the final electron carrier we need to be concerned about is coenzyme Q. This is a non-protein, low molecular weight carrier that is mobile in the lipid bilayer of the inner membrane. Moving on to figure 25.9, page 1011. In this figure, we see inside the inner mitochondrial membrane, and we see the carriers are clustered into 3 complexes, each acting as a proton pump that expels hydrogen ions. As the 3 proton pumps pass electrons from one carrier to the next, they also move protons from the matrix into the space between the inner and outer mitochondrial membranes. As protons flow back into the mitochondrial matrix through the hydrogen ion channel and ATP synthase, ATP is synthesized. The first proton pump is the NADH dehydrogenase complex. The second proton pump is a cytochrome BC1 complex. And the third proton pump is a cytochrome oxidase complex. Moving on to the next slide, cellular respiration will generate either 30 or 32 ATP molecules for each molecule of glucose catabolized. And we can see this reaction outlined in this figure below. Recall that 4 ATP molecules are produced via substrate-level phosphorylation, 2 from glycolysis and 2 from the Krebs cycle. So if the 4 ATP produced via substrate-level phosphorylation are added to the 26 or 28 ATP produced via oxidative phosphorylation, a total of either 30 or 32 ATP is generated for each molecule of glucose catabolized during cellular respiration. So this cartoon is on page 1012, figure 25.10, and it summarizes the principal reactions of cellular respiration. Except for glycolysis, which occurs in the cytosol, all other reactions of cellular respiration will occur within the mitochondria. So this is table 25.1 on page 1011 of your book. So I will let you review this table on your own time. And this next slide is the same table. Please review this table on your own time. Moving on to the next slide. Glucose not needed immediately is stored as glycogen. The process that stores it is called glycogenesis. Then, when ATP is needed for body activities, stored glycogen is broken down by a process called glycogenolysis. So looking at figure 2511 on page 1012, we can see glycogenesis and glycogenolysis. The glycogenesis pathway converts blood glucose into glycogen, and the glycogenolysis pathway breaks down glycogen into glucose. Furthermore, glucose may be formed from proteins as well as the glycerol portion of triglycerides, lactic acid, and certain amino acids. This process is known as gluconeogenesis. Cortisol, glucagon, and thyroid hormones stimulate gluconeogenesis. And now we're looking at figure 25.12 on page 1013. Gluconeogenesis is the conversion of non-carbohydrate molecules such as amino acids, lactic acids, and glycerol into glucose. About 60% of the amino acids in the body can be used for gluconeogenesis. Moving on to lipid metabolism, and this is on page 1013 of your book. Because most lipids are nonpolar, or in other words, hydrophobic, they do not dissolve in water. Because blood plasma is over 90% water, lipids must be transported combined with proteins produced by the liver and intestines. These are called lipoproteins. And here at figure 25.13 on page 1014, we see a cartoon of a lipoprotein. In this figure, we can see a single layer of amphipathic phospholipids, cholesterol, and proteins surrounding a core of nonpolar lipids. Moving on to the next slide, there are 4 classes of lipoproteins. Chylomicrons, which transport dietary lipids to adipose tissue. Very low-density lipoproteins. These transport triglycerides from hepatocytes to adipocytes. Low-density lipoproteins. They carry about 75% of the total cholesterol in the blood and deliver it to cells. And then high-density lipoproteins, they remove excess cholesterol from the body's cells and the blood and transport it to the liver for elimination. Moving on to the next slide, cholesterol comes from some foods such as eggs, dairy, organ meats, but most is synthesized by the hepatocytes. Increases in the total cholesterol levels are associated with a greater risk of coronary artery disease. Exercise, diet, and certain drugs are used to reduce high cholesterol levels. Furthermore, lipids may also be oxidized to produce ATP. If the body does not need lipids at any given time, they get stored in adipose tissues. Some are used as structural molecules or to synthesize other essential substances. Moving on to the next slide, adipose tissue is used to remove triglycerides from chylomicrons and VLDL. Remember, VLDL stands for very low density lipoproteins. VLDL triglycerides constitute about 98% of all the body's energy reserves. Lipid catabolism, or lipolysis, is the process of splitting triglycerides into fatty acids and glycerol. Lipolysis is catalyzed by enzymes called lipases. Also, epinephrine and norepinephrine can enhance triglyceride breakdown into fatty acids and glycerol. On the other hand, lipid anabolism or lipogenesis is the process of synthesizing lipids from glucose or amino acids. This occurs when individuals consume more calories than needed. So excess dietary carbohydrates, proteins, and fats all have the same fate. That is, they are converted into triglycerides. And this cartoon is figure 25.14 on page 1016. This shows the pathways of lipid metabolism. Glycerol may be converted to glyceraldehyde 3-phosphate, which can be converted to glucose or enter the Krebs cycle for oxidation. Fatty acids undergo beta oxidation and enter the Krebs cycle via acetyl coenzyme A. And here in this cartoon, we can see glycerol and fatty acids are catabolized in separate pathways. Moving on to protein metabolism. Protein metabolism begins on page 1017 in your book. Digested proteins are broken down into amino acids which are not stored but are either oxidized to produce ATP or used to synthesize new proteins. Many proteins function as enzymes. Some are involved in transportation, serving as antibodies, clotting blood, being hormones, or being a part of muscle fibers. Protein catabolism, or the breaking down of proteins, yields amino acids which are converted to other amino acids, fatty acids, ketone bodies, or glucose. Cells oxidize amino acids to generate ATP via the Krebs cycle. And then the synthesis of proteins, or protein anabolism, new proteins are created by bonding together amino acids on ribosomes. So here we see figure 25.15 on page 1018. And in the yellow boxes, we can see the points at which amino acids enter the Krebs cycle for oxidation. Before amino acids can be catabolized, they must first be converted into various substances that can enter the Krebs cycle. Moving on to key molecules at metabolic crossroads, this is on page 1019. Of the thousands of different chemicals in cells, glucose 6-phosphate, pyruvic acid, and acetyl coenzyme A are extremely important in metabolism. Moving on to the next slide, glucose 6-phosphate is involved in the synthesis of glycogen, the release of glucose into the bloodstream, the synthesis of nucleic acid, and glycolysis. So what happens is, shortly after glucose enters a body cell, a kinase converts it to glucose 6-phosphate. So regarding the synthesis of glycogen, when glucose is abundant in the bloodstream, as it is just after a meal, a large amount of glucose 6-phosphate is used to synthesize glycogen. And glycogen, again, is the storage form of carbohydrates in animals. The subsequent breakdown of glycogen into glucose 6-phosphate occurs through a slightly different series of reactions. Synthesis and breakdown of glycogen occur mainly in skeletal muscle fibers and in hepatocytes. Regarding the release of glucose into the bloodstream, if the enzyme glucose phosphatase is present and active, glucose 6-phosphate can be dephosphorylated to glucose. Once glucose is released from the phosphate group, it can leave the cell and enter the bloodstream. Hepatocytes are the main cells that provide glucose to the bloodstream in this way. Regarding the synthesis of nucleic acids, glucose 6-phosphate is the precursor used by cells throughout the body to make ribose 5-phosphate. Ribose 5-phosphate is a 5-carbon sugar that is needed for the synthesis of RNA and DNA. The same sequence of reactions also produces NADPH. NADPH is a hydrogen and electron donor in certain reduction reactions, such as the synthesis of fatty acids and steroid hormones. And finally, glycolysis. Some ATP is produced anaerobically via glycolysis, in which glucose 6-phosphate is converted to pyruvic acid. Pyruvic acid is another key molecule for metabolism. Most body cells can carry out glycolysis. Moving on to the next slide, pyruvic acid is involved in the production of lactic acid, the production of alanine, and gluconeogenesis. Each 6-carbon molecule of glucose that undergoes glycolysis yields 2 3-carbon molecules of pyruvic acid. This molecule, like glucose 6-phosphate, stands at a metabolic crossroads. Given enough oxygen, the aerobic reactions of cellular respiration can proceed, and if oxygen is in short supply, anaerobic reactions can occur. So as I said, pyruvic acid is involved in the production of lactic acid. When oxygen is in short supply in a tissue, as in actively contracting skeletal or cardiac muscle, some pyruvic acid is changed to lactic acid. The lactic acid then diffuses into the bloodstream and is taken up by hepatocytes, which eventually convert it back to pyruvic acid. Pyruvic acid is also involved in the production of alanine. Carbohydrate and protein metabolism are linked via pyruvic acid. The transamination of an amino group can either be added to pyruvic acid to produce the amino acid alanine or be removed from alanine to generate pyruvic acid. And pyruvic acid is also involved in the gluconeogenesis. This is where pyruvic acid and certain amino acids can be converted into oxaloacetic acid, one of the Krebs cycle's intermediates, which in turn can be used to form glucose 6-phosphate. This sequence of gluconeogenesis reactions bypasses certain one-way reactions of glycolysis. Moving on to the next slide, acetyl coenzyme A is involved in helping 2-carbon acetyl groups enter the Krebs cycle, and it's also involved in the synthesis of lipids. When the ATP level in a cell is low but oxygen is plentiful, most pyruvic acid streams towards the ATP-producing reactions of the Krebs cycle and the electron transport chain via conversion to acetyl coenzyme A. Acetyl-CoA is the vehicle for 2-carbon acetyl groups to enter the Krebs cycle. The oxidative Krebs cycle reactions convert acetyl-CoA to carbon dioxide and produce the reduced coenzymes NADH and FADH2 that transfer electrons into the electron transport chain. Oxidative reactions in the electron transport chain in turn generate ATP. Most fuel molecules that will be oxidized to generate ATP, that is glucose, fatty acids, and ketone bodies, are first converted to acetyl-CoA. In the synthesis of lipids, acetyl-CoA can also be used for synthesis of certain lipids, including fatty acids, ketone bodies, and cholesterol. Because pyruvic acid can be converted to acetyl-CoA, carbohydrates can be turned into triglycerides. This metabolic pathway stores some excess carbohydrate calories as fat. Mammals, including humans, cannot reconvert acetyl-CoA to pyruvic acid, so fatty acids cannot be used to generate glucose or other carbohydrate molecules. This figure is on page 1019, and it is figure 25.16. This figure summarizes the role of key molecules in metabolic pathways. The double-headed arrows indicate the reactions between two molecules that may proceed in either direction if the appropriate enzymes are present and the conditions are favorable. And then the single-headed arrows signify the presence of an irreversible step. So 3 molecules, glucose 6-phosphate, pyruvic acid, and acetyl coenzyme A, stand at the metabolic crossroads. They can undergo different reactions depending on your nutritional or activity status. And this table is on page 1021, table 25.2. And as usual, I will let you go through the table on your own time. This next slide is a continuation of the same table. Please review this. And this table is a continuation of the same table. Please review this. Again, here we're looking at the same table. Please review this on your own time. I do like to pull quiz and exam questions from the tables. Metabolic adaptations begins on page 1020. So the regulation of metabolism depends on chemicals in the cells and signals from the nervous and endocrine systems. Some aspects of metabolism depend on the time elapsed since the last meal. During the absorptive state, glucose is readily available. During the absorptive state, some of the absorbed nutrients are catabolized for the body's energy needs, or some are used to synthesize proteins. So if we turn the page to 1022 and look at figure 25.17, we see the principal metabolic pathways during the absorptive state. During the absorptive state, most body cells produce ATP by catabolizing glucose to carbon dioxide and water. If we look at number 1 there in the upper right-hand corner, we see the catabolism of glucose. Number 2, down in the liver, we see the catabolism of amino acids. And number 3 is also, but protein synthesis really takes place in virtually every cell, but that is attributed to number 3. And number 4 is the catabolism of a few dietary lipids. 5 is glycogenesis. 6 is lipogenesis. And 7 is the transport of triglycerides from the liver to the adipose tissue. And this next slide is table 25.3 on page 1023. I will let you review this table on your own time. Same table continued. During the post-absorptive state, energy needs are met by fuels already in the body. And if we turn to page 1024 and look at figure 25.18, we see the principal metabolic pathways during the post-absorptive state. The principal function of the post-absorptive state reactions is to maintain a normal blood glucose level. So if we look more closely at this cartoon, we see glycogenolysis in the liver at number 1. Number 2 is pointing out glycogenolysis in the muscles. Number 3 is pointing out lipolysis. Number 4 is showing you protein catabolism. And number 5 is showing us gluconeogenesis. Number 6 points out the catabolism of fatty acids. Number 7 points out the catabolism of lactic acid. Number 8 points out the catabolism of amino acids. And number 9 points out the catabolism of ketone bodies. And this table is on page 1025, table 25.4. It summarizes the hormonal regulation of the metabolism in the post-absorptive state. So please review this slide on your own time. So during fasting and starvation, the body must make metabolic changes to survive. Fasting is defined as going without food for several hours or a few days. And starvation is defined as going without food or inadequate food intake for weeks or months. However, the most dramatic metabolic change occurring with fasting and starvation is an increase in the production of ketone bodies as the catabolism of fatty acids increases. Ketone bodies may be used for energy by all cells.
I've mentioned ketone bodies a few times now, and they are produced by the liver and used peripherally as an energy source when glucose is not readily available. Moving on to the next slide, energy balance on page 1025 is where we're at in the book. Energy balance refers to the precise matching of energy intake to energy expenditure over time. The rates of metabolic reactions control the amount of heat produced by the body. The rate of heat loss must equal the rate of heat production to maintain homeostasis of body temperature. The metabolic rate is the overall rate at which the metabolic reactions use energy. Metabolic rate is measured with the body in a quiet, resting, and fasting state. This is referred to as the basal metabolic rate. Moving on to the next slide, some factors that affect metabolic rate include exercise, hormones, the nervous system, body temperature, ingestion of food, age, gender, climate, sleeping, and malnutrition. During strenuous exercise, the metabolic rate may increase to as much as 15 times the basal rate. Thyroid hormones are the main regulators of the basal metabolic rate. During exercise or in a stressful situation, the sympathetic division of the autonomic nervous system is stimulated. The higher the body temperature, the higher the metabolic rate. The ingestion of food raises the metabolic rate 10 to 20% due to the energy costs of digesting. The metabolic rate of a child in relation to its size is about double that of an elderly person due to the high rates of reactions related to growth. And concerning the other factors that affect metabolic rate, including gender, which is lower in females except during pregnancy and lactation, climate, which is typically lower in tropical regions, and sleep, which lowers the metabolic rate, and of course malnutrition can also lower the metabolic rate. Moving on to the next slide, heat is transferred from the body to the environment via conduction, which is the heat exchange that occurs between molecules of two materials that are in direct contact with one another. Convection is the transfer of heat by the movement of air or water between areas of different temperatures. The contact of air or water with your body results in heat transfer. Radiation is a transfer of heat in the form of infrared rays between a warmer object and a cooler one without physical contact. And evaporation is the conversion of a liquid to a vapor. Moving on to the next slide, the preoptic area of the hypothalamus is the body's thermostat. Thermoreceptors send information to the preoptic area, which sends signals to the heat-losing center and heat-promoting center of the hypothalamus depending on the body's needs. Furthermore, negative feedback mechanisms conserve heat and increase heat production. And on this slide, we see figure 25.19 on page 1031. This is showing the negative feedback mechanisms that conserve heat and increase heat production. Core temperature is a temperature in the body structures deep to the skin and subcutaneous tissue. Shell temperature is a temperature near the body's surface. Moving on to the next slide. Energy intake is directly dependent on the amount of food consumed. The total energy expenditure is based on the basal metabolic rate, which comes in at 60%, physical activity, which comes in at 30 to 35%, and food-induced thermogenesis, which accounts for the remaining 5 to 10%. Moving on to the next slide, the arcuate nucleus and the paraventricular nucleus of the hypothalamus are the areas that control hunger. The hormone leptin helps to decrease adiposity, or the body's fat mass. Neuropeptide Y stimulates food intake. And melanocortin inhibits food intake. So moving on to nutrition, and this starts on page 1032 in your textbook. Nutrients are chemical substances in food that body cells use for growth, maintenance, and repair. The 6 main types of nutrition are water, carbohydrates, lipids, proteins, minerals, and vitamins. Water is needed in the largest amount, about 2 to 3 liters per day. As the most abundant compound in the body, water provides the medium in which most metabolic reactions occur, and it also participates in some reactions like hydrolysis reactions. Minerals are inorganic elements that occur naturally in the Earth's crust. In the body, they appear in combination with one another, in combination with organic compounds, or as ions in solution. Also, organic nutrients required in small amounts to maintain growth and normal metabolism are called vitamins. Unlike carbohydrates, lipids, or proteins, vitamins do not provide energy or serve as the body's building materials. Most vitamins with known functions are called coenzymes. So this is table 25.6 on page 1026 of your textbook. And so we define a calorie as the amount of energy in the form of heat required to raise the temperature of 1 gram of water 1 degree Celsius. Because the calorie is a relatively small unit, the kilocalorie is often used to express the energy content of the foods that we eat. So please review this table on your own time. And this table appears on page 1028. This is table 25.8. And this talks about certain activities that you can do and approximately how many kilocalories per hour this may burn. So review this table on your own time, and I would say take it with a grain of salt, because if you're slow dancing or if you're swing dancing, it's probably going to affect the amount of calories you're burning. So just keep that in mind. This next slide is also a continuation of table 25.8. And this table here is on page 1034, table 25.9. So you can see that calcium is the most abundant mineral in the body. So I'll let you go through this table on your own time. Two of 8. 3 of 8. 4 of 8. 5 of 8. 6 of 8. 7 of 8. And 8 of 8. And now this next slide brings us to table 25.10 on page 1035 in your book. And this table outlines the principal vitamins. So you can see that there's basically 2 types of vitamins, and that's fat-soluble and water-soluble. So again, I'll let you go through this table on your own time, but here's 1 of 9. 2 of 9. 3 of 9. 4 of 9. 5 of 9, 6 of 9, 7 of 9, 8 of 9, and 9 of 9. And this brings us to homeostatic imbalances. In anorexia nervosa, this is a chronic disorder characterized by self-induced weight loss, negative perception of body image, and psychological changes that result from nutritional depletion. Fever is an elevation of the core body temperature caused by a resetting of the hypothalamic thermostat. The most common causes of fever are viral or bacterial infections and bacterial toxins. Other cases are ovulation, excessive secretion of thyroid hormones, tumors, and reactions to vaccines. And obesity is a body weight more than 20% above the desirable standard due to excessive accumulation of adipose tissue. More than 1/3 of the adult population in the United States is obese. Even moderate obesity is hazardous to the health. It is a risk factor in cardiovascular disease, hypertension, pulmonary disease, non-insulin dependent diabetes mellitus, arthritis, certain cancers, varicose veins, and gallbladder disease. And that concludes our lecture for this evening.